In this video, we're going to focus on the Krebs cycle. So just to review, glycolysis is a process that splits glucose into two pyruvate molecules. Now, following pyruvate oxidation, that's when pyruvate is oxidized to acetyl-coenzyme A. Acetyl-coenzyme A enters the Krebs cycle, where it reacts with oxaloacetate to produce citrate. The enzyme that catalyzes that reaction is citrate synthase. It synthesizes citrate from this 4-carbon molecule, oxaloacetate, and the 2-carbon acetyl part of acetyl-coenzyme A. So we get a 6-carbon molecule. Now, another enzyme that I would like to focus on is the dehydrogenase enzyme. Notice what this enzyme does. It removes hydrogen from molecules. Let's focus on step 5. Starting with isocitrate, notice the number of hydrogen atoms that it has. So we have 1, 3, 4, 5, 6, 7, 8. Now, if we look at alpha-ketoglutarate, we can see that it has 1, 3, 5, 6 hydrogen atoms. So whenever you see the enzyme dehydrogenase, it typically removes hydrogen from a molecule. NAD+ then picks up those hydrogen ions as well as electrons, reducing into NADH. Now, whenever there's a reduction reaction, there is an oxidation reaction. Isocitrate has 6 carbon atoms, and alpha-ketoglutarate has 5 carbon atoms. Notice that we lost a carbon in the form of CO2. So step 5 was a decarboxylation reaction. We lost carbon dioxide. A decarboxylation reaction is an oxidation reaction, which involves the loss of electrons. NAD+ picked up 2 electrons and a hydrogen ion, turning it into NADH. So NAD+ is reduced to NADH because it received electrons, while isocitrate was oxidized to alpha-ketoglutarate because it lost electrons. So keep that in mind. Oxidation involves a loss of electrons. Reduction involves a gain of electrons. In step 6, we have the use of a dehydrogenase enzyme. This is alpha-ketoglutarate dehydrogenase. And we're going to take away hydrogen from alpha-ketoglutarate to make succinyl-CoA enzyme A. So we have a total of 6 hydrogen atoms there. In succinyl coenzyme A, there's 5. So we lost a hydrogen, and that was transferred to NAD+ to make NADH. We also got another CO2 molecule, so that's also a decarboxylation step. So the 2 carbon atoms in the acetyl part of acetyl coenzyme A were converted into carbon dioxide, because that's the net result of cellular respiration, converting glucose into carbon dioxide and water. Now, in step 7, as succinyl coenzyme A converts into succinate, we see that a GDP molecule is converted to GTP. Now, GTP converts back into GDP, and in that process, it's not shown in this diagram, but ADP will pick up the phosphate group from GTP, turning into ATP. So we get 1 molecule of ATP in step 7. This part is a cyclic process. We use up GDP to make GTP, and then GTP transfers the phosphate group to ADP to make ATP. In step 8, we have another dehydrogenase enzyme, which means that we're going to remove hydrogen from succinate to make fumarate. So succinate has 6 hydrogen atoms, fumarate has 4, and we can see that those 2 hydrogen atoms were transferred to FAD. Anytime you transfer hydrogen atoms to a molecule, you're reducing that molecule. So FAD was reduced to FADH2, and succinate was oxidized to fumarate. Now let's summarize the Krebs cycle. So pyruvate has 3 carbon atoms. In the first step, it loses a carbon in the form of CO2, but that's pyruvate oxidation. But once we start with the Krebs cycle, that is once acetyl-CoA enzyme A enters it, those two carbon atoms are converted into 2 molecules of CO2. And notice that we get a total of 3 NADH molecules. So one turn of the cycle produces 3 NADH molecules. We also get 1 FADH2, and we get 1 molecule of ATP. So that's for one turn of the Krebs cycle. Now, glucose will generate 2 pyruvate molecules, which turns into 2 acetyl-coenzyme A molecules. So one glucose molecule is equivalent to 2 turns of the Krebs cycle. So one glucose molecule will yield 6 NADH molecules, 2 FADH2 molecules, and 2 ATP. So that's the net result of the Krebs cycle for one molecule of glucose. You get 6 NADH, 2 FADH2, and 2 ATP molecules. So just keep that in mind. Now, out of the 6 carbon atoms that are in glucose, 2 of them are lost in pyruvate oxidation. Each pyruvate loses 1 CO2 molecule, so glucose loses 2 CO2 molecules during this step, pyruvate oxidation. The other 4 CO2 molecules are lost during the 2 turns of the Krebs cycle. So to review, the Krebs cycle is a cyclic process by which acetyl-CoA enzyme A, the acetyl part, those 2 carbon atoms are oxidized into CO2. And during that oxidation process, electrons are stripped from the acetyl group and transferred to molecules such as NAD+ and FAD to produce NADH and FADH2, which will later go into the electron transport chain to make even more ATP molecules. So that's the basic idea behind the Krebs cycle. We're just oxidizing the acetyl group into carbon dioxide, producing these high energy electron carriers.